everyone. Well, thank you to the few that are still around for Thanksgiving week. I asked Ben if he chose this week intentionally to have a small audience as possible. The answer was no, so that's good. So it is my pleasure to introduce Ben Moran today. Ben is a postdoc that just started in my lab. Before he came here, he started his undergrad at Northeastern, where he started working in evolution and marine systems in a couple of different labs, and then went on to do his PhD in Molly Schumer's lab down at Stanford. Overall, his research program is really interested in the origin and maintenance of biodiversity, and he's done some really beautiful work, which he'll show you today, looking at how species arise and how they're maintained and when that breaks down. The thing I really like about Ben's work is that he is unafraid of drawing on different disciplines and different approaches to really dig down deep on the question. And we've been lucky to recruit him here. He's working with Schmidt.
Lucky now.
He managed to get a Schmidt Science Fellow, and we've been lucky to recruit him to work in a less tractable system. So we're really excited about that. But yeah, I'll let him tell you about his work.
Cool. Thank you. Yeah, I did not. I'm glad to be talking to you all. Doesn't matter if it's the few, the crowd, whatever, it's good. And yeah, I have not actually done that much yet in the Bay Lab. So I'll be talking basically about a lightly edited version of my exit seminar from my time at Stanford when I was working on disturbance and dysfunction in swordtail fish hybrid zones. And as Rachel alluded to, kind of the main, also there's it's way over the top for a CPB seminar because it is my exit seminar. So the main question, both in that lab and then also that I was interested in and continue to be interested in, is how species diverge in the first place. What are the processes that create the physical and genetic diversity that we see? Because we might look at an ecosystem and see in these swordtail fish that there are quite a few different phenotypes that are present, and this sort of discrete, diverse phenotypes that we see implies that there's divergence in the DNA. But in order for there to be divergence in DNA of groups in sympatry, that implies that there's some sort of barriers in place that are keeping them from mating with each other. Otherwise, their DNA would homogenize and they would not be physically different. But the question of what actually creates these barriers and allows for DNA to diverge to create these really fun different phenotypes and different ecologies is one that we're still actively figuring out. What barriers matter the most, which ones evolve first, things along those lines. And to talk about two kind of broad categories that will come up in this talk. There's tons of different barriers to the flow of genes. We might think about barriers that happen before mating, such as mating preference. So we might have females of a species that choose their mates based on phenotypes, and based on their genetics, they might prefer one phenotype and reproduce with that one, whereas they don't like the other phenotype and they refuse to mate with it. There's no gene flow, the phenotypes stay separate. On the other hand, we can have barriers that happen after mating, a subset which we might call hybrid fitness. So we can imagine that maybe the female doesn't really care that much. She'll mate with whoever, but due to some differences in the biology of these organisms, once they reproduce, there are differences in the number, the viability, the fecundity of those offspring that are reproduced. And so even though we have mating, the effective gene flow between the groups is decreased. And if these hybrid fitness is low enough, then it can be essentially zero gene flow between the groups, even if mating is happening. And I was interested in studying these in hybrid zones as a kind of counter example to the idea of these reproductive barriers. It's hard when the barriers are so strong that you never actually see them in action. It's really hard to understand them. But in hybrid zones, we can look out and see all sorts of different combinations of phenotypes and genotypes that allow us to test which of these differences are actually acting to keep species separate. So from a genomic perspective, in a hybrid zone, we reshuffle the two ancestral genomes of the species that are present, creating all these sorts of different combinations of phenotypes that are linked to different genotypes. And by just surveying the population writ large, we can start to develop genotype-phenotype associations that tell us about the root of the traits in the population, but also which ones are creating these barriers to reproduction. So we might, instead of just mapping the fin size, we can also map which parts of the fin size are actually, or other traits, I'm just saying the fin size so you can see it, is important for the female's choice and map that genetically. We could also map when she does reproduce which males she reproduces with more successfully. And then by doing that many times, we can understand where the barriers to reproduction are found within the genome. And of course, this is interesting from understanding the sort of traits and the reproductive barriers that are present. But I should also point out that hybridization is an evolutionary force that matters in the course of evolution. There are more and more cases known across pick your own clade. There's hybridization happening. There's a coral up there. And that was on there before I started working in a coral lab, for the record. But it's shaping evolution by the flow of alleles, adaptive introgression, things along these lines. But it's also happening more often, which I will talk about a lot in the recent past, we believe, due to changes in the environment caused by people. And in certain vulnerable taxa, hybridization actually presents a threat to their existence as a sort of independent genetic lineage. And so understanding how hybridization works is both important for understanding the course of evolution as well as the persistence of taxa in the Anthropocene. I studied these questions in swordtails, or fishes of the genus Xiphophorus. They're found in kind of all the drainages that lead into the Gulf of Mexico. I studied in particular 2 species found in the state of Hidalgo, Mexico. In the eastern Sierra Madre, Xiphophorus malinche, which is found in the highlands of these mountains, and Xiphophorus birchmani, which is found in the lowlands, and where they overlap at intermediate elevations, we see hybrid zones between them. And what's fun about freshwater systems is that each of these rivers is on ecological and sort of demographic timescales is more or less independent. And so we have replicates, per se, in a natural experiment, trying to understand the course of evolution during hybridization. We can compare between different rivers, see what patterns hold across rivers, what varies, and then understand if there's differences between the rivers that drive that, or if it's just kind of free playing the tape of evolution and getting different outcomes. And so the central questions that I tried to answer in this Xiphophorus malinche birchmani hybrid system was, first, on the mating preference side, can environmental change break down behavioral barriers to hybridization, such as mating preference? And on the hybrid fitness side, the post-mating barriers, what cellular and molecular processes impact hybrid fitness the most in these natural hybrid zones? Like, what is actually the strongest barriers keeping these species apart in the wild? So to go into answering part 1, talk a little bit about anthropogenic disturbance and hybridization in these swordtails, which is a manuscript that went out for review a couple weeks ago, which means I'm waiting for the other shoe to drop, which is terrifying, but it's fine. Fingers crossed for easy reviews, probably not, but such is life. So I alluded to before that hybridization is increasing. There's a lot of taxa where we know, based on genetic estimates or just strong historical records, that the hybridization didn't start happening until, say, the 1900s or even the 2000s. And people have been thinking a lot about this concept of anthropogenic hybridization and placing it in a broader context of sort of disturbance in the environment and how that affects signaling between species. So if we think about how species are choosing mates, they're observing each other, seeing some sort of phenotype or perceiving some sort of phenotype if it's not vision, and then making mate choices based upon that. Any change to the environment can disrupt the signal that's being presented, the transmission of that signal, or the reception of the signal. So if we have, for example, a pollution of the environment by people, basically, it can disrupt the sensory systems of these organisms, change their decision-making processes, and cause hybridization to happen where it wouldn't happen before. I was particularly interested in whether the strength of disturbance could affect the evolutionary outcomes in each scenario. So we can imagine a case where we have 2 species with our little ancestry histograms here, so they're nice and separate, nothing in between, and then we introduce some sort of disturbance that causes early generation hybrids to be formed. Now we can imagine one scenario where gene flow is rare and weak relative to the strength of selection against hybrid ancestry. And in this case, we might see what we call a stable hybrid zone, where we continue to observe early generation hybrids, but we also maintain this sort of bimodal ancestry structure, and sort of distinct ancestry clusters, what you might call them. And we assume we're kind of homogenizing the neutral parts of the genome, but maintaining the ecological diversity of the system. On the other hand, if there's so much gene flow that it overwhelms any selective pressures against hybridization, we can create what we'd call a hybrid swarm, this unimodal population that at the beginning is going to have pretty high variance in ancestry. And this is obviously different for the evolutionary outcomes because we've gone from one lineage or two lineages to one. And then also if we're thinking about it in terms of conservation, people generally have the presupposition that more diversity is better. Here we feel like we're losing a taxa, and the first one we don't feel like we're losing a taxa. So there's also a sort of like qualitative difference in the way we think about these things from a conservation perspective. Going back to the swordtails, I mentioned that this hybrid zone exists. What I didn't mention is that both historically and genetically, we estimate it started about 35 years ago, which is a little bit suspicious given that we think they've overlapped for on the order of 200,000 years. Or at least they diverged 200,000 years ago. Who knows how long sympatry was, but it's very recent relative to the scale of the sympatry. And this implies that something changed in their environment that would lead them to them starting to hybridize. And there's plenty of hypotheses of what that change might be because there's a lot of ecotoxicology literature on how water chemistry can affect fish's mate choice. In these hybrid, or in these Xiphophorus, I mentioned that they have both visual signals, so they have their big fins and their swords and their pretty colors, that is being displayed by courtship behavior that is perceived by the female. They're also producing pheromones that are transmitted through the water to the female's olfactory system and then processed by the brain. And any of these steps could hypothetically be disrupted by water pollution. So we know pharmaceuticals that can disrupt both behavior and the production of pheromone signals through like endocrine disruption. We also can imagine turbidity affecting vision. We could have organic matter in the water that sequesters organic pheromones that are present. And we can also know that heavy metals in the water are also sometimes neurotoxic and can affect the olfactory epithelium directly. So in general, in fish, we know that water pollution can affect mate choice quite strongly. In these fish in particular, these Xiphophorus, there are also studies that have been done showing that the pollutants that we find in the wild for these fish can ablate or even reverse pheromone-based mate choice specifically. So people have taken these females, introduced dissolved organic matter, bumped up the pH and the calcium through slaked lime, which they chose because people purposefully dumped that in the river in this area, and shown that that will ablate pheromone-based mate choice in these species. So we already have experimental evidence that this might matter, but we don't actually know what's going on in the water that they're living in, whether this tracks with where we're seeing a hybridization. The inspiration for this particular study was in the town of Kaunali, which is kind of the municipal seat for the area. It's about 3,000 people and it's agrarian landscape. And we see this really cool pattern. We had preliminary evidence where upstream in the headwaters of this is the Kaunali River going upstream to downstream. In the upstream, we see pure-ish Xiphophorus malinche. We move a little bit downstream and we see what we might call a stable hybrid zone where they've been sampling for 20-ish years now. Well, 15, so 30 generations of fish, and the ancestry structure looks pretty much like this all the time. There are always two clusters with maybe a few intermediate individuals, but they've stayed at roughly the same ancestry proportions over time. So we think that there's like a gene flow selection balance happening. But when we sampled downstream of the town more recently, we realized that downstream of the town of Kaunali, there was a hybrid swarm present. And it's a mix of phenotypes matched from both species, and also more variable ancestry. And so this implies that something's happening in the town, because this is a very short distance. This is like a kilometer. So they're demographically connected, but we see structure upstream, but not downstream, implying that one of the various point source pollution sources in the town might be affecting the water quality enough that it is driving a change in the reproductive barriers between these species. And so my hypothesis here was that water pollution is driving a collapse of species barriers between Xiphophorus malinche and Xiphophorus birchmanni. And I tested this sort of chain of causation that you can imagine starting with, all right, there are more humans in the area is first, if we believe that humans are causing the hybridization, then we would hypothesize that there are more humans in the places where we see hybridization. Those humans cause a change in the environment of the fish, which I'm going to quantify through water chemistry, that then disrupts the olfactory systems of the fish, which we believe that olfactory choice is more important for between species discrimination rather than visual being more important for within species discrimination. We disrupt the olfaction, the females lose preference, and we see a loss of assortative mating by ancestry that then leads to the collapse of population structure. So this is kind of the causal chain we're hoping to infer. And then I did a bunch of different analyses and assays that I'm going to step through here to try and at least get some idea of each of these steps, if not prove them. And I did this process in 4 rivers. Again, butterfingers. 4 different rivers, all within the Rio Atlapeshco drainage. The Huasalingo, the Calnali that I just talked about is our fun, like, central river, the Pochula and the Concitla. So we're hoping that these are not just demographically isolated, but also different in their anthropogenic impact, such that we might be able to relate variation in anthropogenic environmental differences to differences in the other downstream processes that we're hoping to see. So to start with, are there hybrids or not? I was very fortunate to inherit an entire dissertation's worth of genomic sequences from the Pochula and the Huasalingo River, and then I did the sequencing, or I collected for the Concitla River, and then through a combination of my fieldwork and some other people's fieldwork, we got Canali River. So we built up 40 plus sites across 4 rivers where we had 19 or more, some between tens and hundreds of samples that we did low coverage genome sequencing followed by HMM based ancestry calling. These species are about 0.5% diverged, so doing like, we can get pretty good resolution from very low coverage data in terms of whether a particular like kilobase of DNA came from one species or another. So doing that across a few 1,000 plus individuals, I'm going to show you graphs that are histograms. So just to show one to orient you, each horizontal histogram here is a site, a sampling site. The y-axis is the individual's ancestry proportion, with 0 being pure Birchmani, 1 being pure Malinche. The width of the bar, since it's sideways, is the number of individuals that fell into that ancestry bin. And so in the Huasalingo, we can see what is like roughly a classic clinal hybrid zone, where we have pure Malinche at the top, pure-ish Birchmani at the bottom, a transition that here actually happens quite dramatically. And there is one site where we see some pseudo sympatry where we detect significant bimodality in the sample where there is Malinche-ish, Birchmani-ish, and a lot of intermediate individuals. Interestingly, this is just downstream of a waterfall. So we imagine like Malinche individuals kind of spilling over the waterfall, and the Birchmani have run up against it and can't get any higher. Fun little demographic aside there. Next river, the Pochula, also has a sort of clinal pattern, except we see more of a gradual change. Interestingly, this one doesn't have a dramatic waterfall at any point. So we see Birchmani ancestry more or less smoothly transitioning into Malinche ancestry as we move downstream to upstream. Now our kind of focal river that inspired this, the Rio Calnali, we see the cool pattern we were hoping for, where at our upmost site, we have pure Malinche. Downstream from that, we have these really nice ancestry clusters, which have a few intermediate individuals, but strong bimodality in the ancestry. And if we look downstream, we see this collapse to a hybrid swarm that then kind of stabilizes in the variance of the ancestry as you go downstream, which is interesting. But it's still just one single population. And of course, in the middle is where the town goes, or where the river goes through the town of Calnali. So that was what we saw based on phenotypes and some preliminary data. So we were excited to see that fleshed out. The final river that I was happy to have bothered to sample because it became sort of our negative control is the Concitla, that has much lower population density than any other river in the area. And here we see, one intermediate individual in the entire river, and really what looks more like an ecological turnover where we have an area of sympatry, and they're not 100% Malinche, Birchmani if we go based on the ancestry calls, but there's very few intermediate individuals. It seems like it's just a turnover when you go from the highland species to the lowland species with this area of sympatry. So this was exciting because we now see, unfortunately, the experiment has no replication because each river is different, but at least we have variation that we can look how that correlates with differences in the water chemistry. So to walk it back now to the very beginning, we have our differences in the hybridization outcomes, and we're interested in how this relates to differences in anthropogenic impacts. So I'm going to start with showing you land use classification from Landsat data. And this is just, so this is that whole watershed again. We have our 4 rivers. Green is forest, yellow cleared land, red is the most interesting one here because that's essentially buildings, developed land. And we can see that the town of Kaunali is kind of the biggest red blob in the whole area, and it's the largest population center that's directly on a river. We can quantify this as the proportion of the watershed upstream of each sampling site that has the proportion of developed land. And sure enough, the Kaunali has this really, oh my gosh, Ben. Take away the button. Has this really exceptional spike in the proportion of developed land relative to the size of the watershed that supports this being a like uniquely impacted river here. But we don't want to just assume that more people means more pollution. So now we do water chemistry. I measured a raft of parameters across a largely overlapping, although partially non-overlapping set of sites in these 4 rivers. N of 3 for most places, N of 2 for a couple where disasters happen, as they tend to do. And I won't walk through each parameter in detail because there's dozens, but I will quickly flash a PCA of just ignore the loadings for now and just look at each river, and then I split the upstream and downstream Kaunali. This is non-metal parameters, and this is mass spec data of ions. And we see surprisingly similar PCAs in each case where we have kind of we see clustering by river and then also clustering of the downstream Kaunali different than the upstream Kaunali. To put this in more, I guess, intuitive terms for me at least, I don't know, we can look at a few representative parameters and how they play out on the map. So these are our 4 rivers again, these are our sampling sites, and in this case, dissolved organic matter, we can see that the lighter colors are higher values, and we see higher values of dissolved organic matter in the Kaunali in general, but especially high downstream of the town. Similar pattern in ammonia, also in nitrite, which I'm not showing. And then in those heavy metals that I mentioned, again, in copper, which is a known neurotoxin in sufficient quantities, we see higher values downstream of the town of Kaunali. Same with cadmium, a raft of other parameters. I should mention that there are ones that are, for example, higher, that they vary, the variance is mostly partitioned by river, not by are you upstream or downstream of the town. And there's some like uranium and manganese that are way higher in the Conzilla than anywhere else, which I have ideas about if people are curious about that later. But it just to say that like we can rule out some potential candidates if we buy this idea that like if we can rule if it's high in the Conzilla and we still see population structure, then that alone would not drive a breakdown in population structure. So it's nice to see that it's not all just higher downstream of the Kaunali. We do eliminate some possible candidates based on this analysis, which is fun. So we show that there's genetic variation, we show there's environmental variation. Now to try and get at the mechanism, I did kind of go immediately towards olfaction here rather than vision because other people have done vision work in this system. And a lot of people have done behavioral experiments, which I did not do for 2 reasons. 1, I did a pilot and it failed miserably. And 2, after that, I was like, well, maybe we could get creative and look at something like part of the phenotype that people haven't looked at before. And so I went with histology, like the actual tissue of the nose, and seeing if that was affected in these cases. So to orient you to a fish nose, they have these structures called olfactory rosettes that are composed of alternating sensory beds with the ciliated neurons that collect like actually binding the chemicals from the environment. And then they are interspersed between non-sensory tissue. There's a lot of connective tissue with some goblet cells that produce mucus as a sort of physical barrier against irritants for these sensitive neurons. And I did not actually make the slides because, that's hard. But I worked with somebody to produce the slides of these olfactory rosettes where we can again see our sensory beds here with our interspersed non-sensory tissue. We can do specific stains for goblet cells and counted the proportion of these ciliated beds that still had their cilia because the cilia can slough off under sort of acute stress. And then also the counts of these goblet cells per area. And I measured these statistics in the Rio Canali at 2 sites, one upstream of the town that we know has lower pollution and one downstream of the town that we know has higher pollution. And working with an awesome undergrad, Terrence, we found that there was a marginally lower level of tissue in the polluted, of cilia in the polluted site. So more cilia loss at the polluted site, and then very significantly higher counts of these goblet cells. So it seems like the goblet cells are proliferating at this downstream polluted site. And these are close enough also that we assume this is like a plastic effect, not an adaptive change. But it's interesting to see that there is, I think the take home, because I don't want to like get too far in front of my skis, but like the take home is at least that there's something different in the environment that the olfactory biology is reacting to. And that's consistent with sort of actually a lot of like EH&S itch literature on what happens in response to olfactory irritants in other vertebrates. So we have a phenotype, which is fun. And then now finally going back to the crux here, which like, is there differences in assortative mating by ancestry between these 2 places? It's very hard to quantify mate choice in the wild by looking at the fish. So instead, we got creative with genomics by inferring the ancestry of mates based on that of mothers and embryos. Swordtails are cool because, well, among other reasons, they're live-bearing fish. So the females carry embryos in an independent evolution of pregnancy. And so we can get a pregnant mother, sequence the mother and the embryos, and see, does the embryo have similar ancestry to the mother, or does the embryo have different ancestry to the mother? And since, you're getting half your DNA from both your parents, we can infer that if the offspring ancestry is similar to the mother, then the father must have had similar ancestry. If it's different, then it must have been different. So in general, as like a relative metric, we know with increasing assortative mating by ancestry, we have decreasing differences between mothers and fry in their ancestry. And so I use this to infer through simulations the relative strength of assortative mating at these different sites. So we start with our real data where we have the mothers and the embryos, and we just calculate our summary statistic, which is the ancestry difference between the mothers and the offspring. And then we do simulations of assortative or random mating, where we pick a strength of assortative mating. I did 2 different models. One that was a sort of stepwise function where it's either you're like assortative or you're not. Here I'm showing a different one that I did, which is a continuous distribution where the farther away your ancestry is, the less likely you are to mate, mostly because it looks prettier when we make the graph. So we can imagine strong assortative mating, where we really don't want to mate with anyone that's much different than us. We can have weaker assortative mating, where there's still a preference for individuals similar to you, but you will mate with most anyone. And then finally, we can have true random mating, where it's just your likelihood of mating with somebody is uniform with respect to their ancestry. And in these simulations, we can pick an assortative mating value randomly. And then once we have it, then take our distributions of ancestry in our population as our like hypothetical group of males and females that are going to mate with each other, simulate mate choice based on this relative preference, and then generate this ancestry, this mother-offspring ancestry difference, and use that to fit which strengths of assortative mating match best with our real data. So first, just comparing our data with simulations of random mating. So here we have red is the downstream polluted site, blue is the upstream cleaner site. And the density plots are the outcomes of average difference in mother-offspring ancestry under completely random mating by ancestry. So this is what the difference would look like given the ancestry fractions at that population if there were no preference for your similar ancestry. Then the vertical lines are what we actually observe in those populations. So in both populations, we see lower differences in mother-offspring ancestry than we would expect under random mating, since we said more assortative mating means lower differences. That implies that there is assortative mating happening in both these populations, which is actually not what we expected. We expected random mating in the like unstructured population, but there are still choosing mates based on ancestry. But you can also maybe notice that the difference between these two is much lower in the downstream population. So we wanted to quantify the strength of this assortative mating. So now I'm going to show the results of approximate Bayesian computation under the sort of stepwise model of ancestry preference that I didn't show. So it's like, if you're similar, you have a certain likelihood of picking them. And if you're not, it goes down by quite a bit. And this value, this P assortative, is just the probability of mating with someone within your cluster. And so we simulated across basically 0 to 1, either it's 0 being complete random mating, 1 being you will not mate with someone outside your cluster. And we found, as we expected, that the cleaner upstream population had near perfect assortative mating by ancestry. The confidence interval includes 1. Whereas the downstream one, it was still there. It's not 0, but it's significantly lower, and it also does not include 1. So again, we're seeing they're still showing preferences by ancestry, but it's much weaker than the upstream site, which is a cool nuance that we didn't expect. So to summarize all that, we have water quality deteriorating at the point where population structure collapses in these rivers. In contrast, in other rivers that we surveyed, we see some where there isn't much ongoing hybridization, and in those rivers, we don't see these sort of dramatic changes in water chemistry that seem to be tied to human development. That we see signs of olfactory damage in a polluted location, and we also see weakening of mating barriers as documented by inferred assortative mating in these places. So that's the first half. And now the second half, which is going on to the, I would say, like the consequences of hybridization, specifically for the fitness of hybrids between Malinche and Birchmanni, which was in the other major wing of the thesis that was in a paper called A Lethal Mitonuclear Incompatibility in Complex 1 of Natural Hybrids, which you might gather from the title, involves mitochondria and being very unhappy when they don't work. But the more theoretical route to this wing is the question of how, again, going back to the beginning, of how species diverge at a genetic level. I think the first sort of naive treatment of how species is like a paradox that doesn't last very long when you really think it through. But like the original paradox was, oh, well, we say everything descended from a common ancestor. But we also, from people who started making hybrids and like around the modern synthesis-y time, and realized that hybrids tend to be not fit. They tend to be inviable. They tend to be sterile. So if these things had a common ancestor, and presumably one genotype evolved to the other, how did we pass through this unfit genotype without being selected against such that the second genotype didn't evolve? And the answer to this is that this paradox goes away the minute you start thinking in more than one dimension of genotype space, like when you involve genetic interactions and epistasis. So now, instead of thinking of one gene, we can think of 2. And I'm going to get a little bit more concrete and show 2 proteins that need to physically interact for function to happen. And in the ancestral alleles of these 2 proteins, we see a proper interaction that does some biological function. Now we imagine that in one species, we see a substitution that changes one of the proteins but preserves the interaction. Then we see a substitution in the second protein that also preserves the interaction. But what we've set up here is that now, if a different species stayed with this original version, we can create a hybrid that has a genotype which has never been, this genotype space has not been passed through in the course of prior evolution. And we don't know whether that interaction is going to work. It hasn't been subject to natural selection before. There's a good chance it disrupts function, it's maladaptive, we decrease hybrid fitness. And this framework of negative epistasis in hybrids, or Dobzhansky-Mueller incompatibilities, or hybrid incompatibilities, is one that's kind of central to the idea of molecular evolution of species barriers. I was interested, because of some preliminary data, in specifically looking at this in mitonuclear interactions. So interactions between genes encoded in the mitochondrial DNA and in the nuclear DNA, which people have highlighted as maybe a hot spot of the evolution of reproductive barriers, largely because there was a lot of evidence showing that hybrids had particularly dysfunctional mitochondria from a physiological perspective. But we also might think that these are a good candidate for the evolution of these barriers for some other reasons, one being the mitochondrial DNA, depending on the taxa, has very rapid substitution rates. So you might get more of these substitutions necessary to create these incompatibilities. And also all these mitochondrial proteins function within large protein complexes where they need to function with nuclear encoded proteins that are imported to the mitochondria. And so there's all of these protein-protein interactions happening, like dozens of proteins within a single complex. If any of them fail, presumably it's possible your mitochondria don't work. If your mitochondria don't work, you don't have ATP, you are dead. And so there's a strong fitness pressure here to maintain these interactions that could be disrupted in hybrids. And so the aim of this part of the thesis was to explore mitochondrial or mitonuclear incompatibilities active in these Xiphophorus-Birchmani hybrids across as many biological scales as possible. So starting with, say, what are the differences in DNA sequence that lead to these incompatibilities existing? What are the genes that are interacting? How does this create individual level dysfunctional phenotypes? And finally, what does this do for broader species level evolution? And again, I am going to breeze through a bunch of different methods from which we explore these things. And I just did it again. Wow. Okay. Starting with, we didn't actually know if there were mitonuclear incompatibilities at the start of this. And so for this, I did what's called admixture mapping. And actually, our Kaunali hybrid swarm here is going to make a reappearance because the large variance in ancestry here is really convenient for mapping in this mitonuclear case. So what we're doing here is like a sister analysis to the GWAS, except our phenotype that we're mapping is your mitochondrial ancestry. So we're looking at if you have in this population both the Birchmani mtDNA and the Malaysia mtDNA are present, and we look at is there a statistical relationship or statistical difference in the frequencies of your genotype ancestry at a particular nuclear locus as a function of your mitochondrial DNA. So if there's no interaction, no association, we expect these two halves to look identical. But in some cases, we see, in particular, depletion of heterospecific combinations. So Malinche mtDNA and Birchmani ancestry might be absent from the population. And that tells us that there's some sort of selective pressure going on that's removing these individuals or favoring the other genotypes that are then proportionally overrepresented. And what's nice about this is that you can put a line through it and do a correlation test. And the strength of your correlation test tells you how strong this selection is. And by doing this at every nuclear locus in the genome, voila, we have a Manhattan plot where the y-axis, the x-axis is the locus that varies between Birchmani and Malinche. The y-axis is the strength of the association. We have a genome-wide significance value based on permutations. And this, we had two significant peaks, but I'm actually going to highlight the third most significant one because the gene in the second one, we don't know what it is. But also the third one in later analyses with larger sample sizes after this paper became significant. So it's not like it's a fake peak. And we were really excited about this because the kind of top candidates in both these peaks, you will notice that their names are very similar. And that's because they're members of the same protein complex, mitochondrial complex 1, which is the first step in the electron transport chain. And within this complex, not only do they interact both with mitochondrially encoded proteins, but they actually interact with each other physically. And so this is a really straightforward candidate for this protein-protein interaction model of mitonuclear incompatibility evolution. So we have our two candidate nuclear genes. We only expected to get one. It's nice. Oh, great, we have two. Awesome. And then now we look and see, all right, what is the DNA sequence evolution that might be causing this incompatibility? So to zoom in on our AlphaFold created structures here and create little semi-transparent bubbles where there's an amino acid substitution, we see a large number of substitutions at the three-way interface between NDUFS5, NDUFA13 in yellow, and this mitochondrially encoded ND6 protein. And it's just like a giant mess of substitutions all in contact with each other, which qualitatively was very exciting. But to make it more concrete, I used CodeML and did branch tests for accelerated evolution and found in particular that Xiphophorus-Birchmani had a really outsized substitution rate within NDUFS5, within NDUFA13, and also within ND6 relative to the other Xiphophorus where we had sequences available for these genes. You see there's higher substitution rates in the mitochondrial DNA in general, but still this was a significant acceleration on the Birchmani branch of evolution here. So where substitutions are kind of the basic currency of these incompatibilities appearing it's nice to see, yes, there's a ton of substitutions here that could be driving differences in function and protein-protein interactions. Now going to individual level dysfunction, I looked at the phenotypes that were present in these incompatible hybrids. Unfortunately, for me being able to phenotype things, the peaks were so strong of the association because all the individuals with the incompatible genotypes were dead. Like there were none. We never observed, specifically with the NUFS5 one, we never observed an adult in the wild, we found one in an aquarium and we think it was a genotyping error out of like 1,000 something genotypes. So it's like a true complete lethal. And so the first question is, can we ever find these incompatible individuals? And so I dissected a bunch of mothers, sequenced their embryos, and this is the embryo developmental stage with kind of pre-fertilization or fertilization here, and then birth over here, genotype and NUFS5 here. All these individuals have Malinche mitochondrial DNA from here on out. So the red Birchmani genotype is the incompatible one. And we see a lot of Birchmani individuals early in development that just completely disappear once we hit a certain developmental stage. So we thought they were like stalling out in development per se, to speak extremely loosely. On the other hand, the NUFA13 genotype, we didn't see any sort of this distortion throughout embryonic development. So when we're at the embryo stage, we see the frequency of this Birchmani genotype is within what we expect from Artie Weinberg, Punnett squares even. But when we hit juvenile, even a few months out, this frequency has started to drop, and by the time we're adults, it's near zero. And so we have, interestingly, even though they're physically in the same complex touching each other, the sort of temporality of the selection is pretty different. So this was good to see. And it also inspired like, well, these incompatible individuals are alive as embryos. So if nothing else, can I like measure phenotypes in embryos and see what's going on here? So I did. I dissected out live embryos and put them through respirometry and also some morphometrics. And I found that the NDUFS5 individuals, do not look great. So these are the normal fish. They have a yolk. And these are siblings, so they're the same age. And this one looks like, it's a fish, roughly. This one is a sad slug on top of a yolk. And they don't get any bigger than that. They just kind of stop. But it is alive. And I put it through respirometry and found that even after controlling for its much smaller size, like controlling for the body volume, there is lower oxygen consumption that's implying lower mitochondrial function in these individuals. And so this aligns with the idea that it is a mitochondrial dysfunction that's driving this. On the other hand, the NUFA13, we did see a difference in respiration, but much weaker difference in respiration. And we also didn't see this dramatic difference in size. They more or less looked like their siblings. But what was really cool was I measured the heart function. You can see their hearts beating because it's inside their yolk. It's external to their body at that point, which is fun. And found that these individuals actually had lower heart rate and increased heart. It's the sinoatrium. It's like the proto-heart. Increased heart size and decreased heart rate relative to their compatible siblings. And this was not that surprising because in kind of other invertebrate mitochondrial dysfunction in general, you tend to see high energy tissues suffer the most. So neurological tissue and muscle and cardiac tissue tend to be the first things to go. So we're seeing signs of altered cardiac function, reduced heart rate, and cardiomegaly in vertebrates is often like it's a form of disease. It's not a good sign. So that all said, we have evidence that it aligns with, yes, this is a mitonuclear problem that's causing bad health effects. And finally, to zoom way out to the sort of species level evolution of this phenomenon, I did a little bit of comparative phylogenetics here. Baby phylogenetics, I'm not a phylogeneticist. But the mitonuclear phylogenetics of swordtails is a mess in that when you look at the nuclear DNA, I didn't mention this, but Malinche and Birchmani are sister species. They group quite nicely, it's very clear. And the sister to them is Xiphophorus cortezi. When you make a mitochondrial phylogeny, we have mitonuclear discordance here, where by sequencing a couple new Malinche populations, I found that cortezi is nested within Malinche diversity, And their mitochondrial DNA is so similar that incomplete lineage sorting is not an explanation here, and it's almost certainly a case of recent introgression between Malinche and Cortezi that replaced one of the mitochondria in one direction or the other. And so this raises the really cool idea where we might have an incompatibility that actually moved between species based on prior hybridization, which is a little bit counterintuitive until you think that like, just because one pair of genes is incompatible tells you nothing about whether it's incompatible with a different allele of that gene. So our sort of pseudo-mathy way of the transitive property says, X Malinche and X Birchmani versions of these are lethal. Malinche and Cortezi are more or less identical, therefore by the transitive property. Zyphophorus Cortezi and Zyphophorus Birchmani of these alleles should have not just lethality, but maybe even a similar phenotype. And I did some preliminary stuff on this in wild populations, but instead I'm going to highlight what I helped a little bit with in a follow-up paper by a postdoc in the lab, where she made Cortezi-Birchmani hybrids and showed that the individuals with Cortezi mitochondrial DNA and Birchmani and UFS5 showed the same sluggy phenotype as the Malinche Birchmani one. So we see the same incompatibility phenotype in the same two genes across two different species, which was very cool to see. So now to summarize part 2 about our consequences here, we had an incompatibility of complex 1, mitochondrial DNA, NDUFS5, and NDUFA13. That was probably driven by accelerated evolution in Birchmani that created this highly divergent contact site between these proteins. And that this lethality is driven by different phenotypes depending on the nuclear gene, either general failure to thrive in mitochondrial dysfunction or specifically effects on the heart. And that finally, that this incompatibility might, or almost certainly introgressed between species in a more ancient introgression event. Okay, that's all the data. So now to walk it back to both parts, I found that this hybrid zone's existence is tied to pollution, and that these mitonuclear incompatibilities are some of the strongest barriers on a hybrid fitness level that are separating these species. And I think a really cool thing that someone else could follow up on later is this whole like shared incompatibility concept. And why do we care about this? Because anthropogenic hybridization is a growing phenomenon that's affecting both evolution in the wild as well as our conservation of biodiversity. And that by doing these integrative studies of hybrid fitness barriers, we can understand more about reproductive barriers in the wild rather than just doing one single level of biological organization. And then finally, this shared incompatibility thing, I think, is a kind of understudied mechanism in evolution. The question of how different hybridization events interacted was a silly question before we understood how often hybridization actually happened. So now that we've recognized that, like, oh yeah, one species might have 3 different hybridization events detectable in its genome, we can start to think about how these phenomena might interact with each other across longer timescales. Yay! Oh, the letter got set. Many thanks. As you imagined, you might have imagined this was extremely collaborative work, and I owe being able to present on this to a lot of people that I'm deeply appreciative of, as well as funders, and I'd be happy to take any questions.
I'm sorry if I missed this.
It was a lot, I'm sorry.
For the hybrids that you see downstream,
Yeah.
where it was like in that really polluted population,
Yeah.
what's their mitochondria?
Yeah, so I will actually zoom back a lot for that because I think it's very fun. Actually, I don't know, it's going to be really obnoxious if I try and like actually just zoom. But I do want to show, I'm going to show I wanted to talk about this in general, so I'm going back. At the upstream sites, they have both, which is what let that admixture mapping happen. So the first site downstream, we see both mitochondrial ancestries present. But as we move downstream, find the slide, there it is, yeah, okay, cool. So as we move downstream here, you notice the ancestry shifting a little bit up. It fixes, so we say for Malinche mitochondrial ancestry. So the Birchmani ancestry goes away. And we would expect this because there's really strong selection happening here. And it's not stable evolutionary. Like there's going to be, it could be arbitrary if the relative fitness of the two is the same, but like stochastically, the population is going to start to lean one way, then whatever the dominant mtDNA is has a fitness advantage, and it's going to really quickly fix in that direction. So the only reason we have both segregating here probably is because we have a constant influx of migrants from upstream.
You saw for your plot of the fitness effects of the incompatibilities that NDUFS like 5 follows more of like a dominant pattern where the heterozygotes are basically not suffering any incompatibility consequences at all, versus 13 follows more of like an additive model. So is the idea that like one of those loci is following a dominance mode of genetic action versus the other is more additive in nature?
Yeah. So it's cool because based on the frequencies in F2s, we could like directly estimate. Oh, there were also F2 hybrids made in the lab by someone else. So based on that, I could infer both the selection coefficient and the dominance coefficient. So both of these had like selection coefficient for NUFS5 is like 0.99. For NUFA13 is 0.9. Oh, I'm starting to forget. I think like 0.97 or so.
Right.
But the dominance for both is actually low. So they're both mostly dominant, or sorry, recessive. They're both mostly recessive. And that is interesting. And I don't quite know how to parse that biologically, except that just like that's what happens.
Yeah.
But yeah, we see the heterozygotes doing mostly fine. Although, there was one slide that I didn't show because this was my defense, and I was trying to focus on things that I actually did. A collaborator took like adult heterozygotes for both, so like double heterozygotes, and put them through mitochondrial respirometry and found that they did have detectably lower complex 1 function. So when they're just chilling out in an aquarium, they're looking fine, but their mitochondria are not. Their complex 1 in particular is not seemingly functioning as strongly. And so we wonder if the not perfect recessivity might be like in dire times, maybe they have like lower endurance or something than individuals that are exactly matching in their mitonuclear ancestry. Yeah. There's also some weird stuff with like the interaction between the two like the 3-way dominance thing.
That's what I was going to ask about next. Is there's obviously interactions between like the mitochondrial protein and then the 2 nuclear ones.
Yeah.
But do you see any evidence of like interactions between the 2 nuclear proteins?
Yes, there was significantly. I don't know if I can actually. I have a slide for this. But is it here? Yeah. Okay. Here it is. And this is getting really wonky. No, what have you done? Okay. Maybe I'll just drag this over because it's a hidden slide, or I can unhide it. Okay, so this is getting really in the weeds here, but it's like, if you are a homozygote for either, you are dead. So that doesn't really matter. So when we're talking about like interactions between the two, the only thing we can really measure is if you're a heterozygote for either, are you more sensitive in the other one? Like, are double heterozygotes worse off? And what we found is that, so if you're a heterozygote for NUFS5, we see that the individuals with Birchman and NUFA13 are even less common than we would expect under an additive model. So it's like, if you are on their own, the two are almost totally recessive.
Yeah.
But if you are heterozygous for the one, and then you happen to be, I mean, it only goes in the one direction, but like if you are heterozygous for NUFS5, and you're homozygous for NUFA13, you are even more screwed than if you were not heterozygous for NUFS5. So it makes the NUFA13 incompatibility go from like very recessive to like it makes it even more lethal if you're already heterozygous at NUFS5. So there is a in the statistical sense, an interaction. Now, how that plays out at the biochemical level, I don't know exactly. It's pretty easy to imagine because they're like physically touching each other, but yeah, hard to say. Yeah, so it's a very in the weeds thing, but the editors got us, or reviewers got really honest about proving that there was an interaction.
Yeah.
And so it's like, there's in the model, there's the interaction term, they're interacting. Yeah, so. Yeah, cool questions.
I'm wondering if you could speculate a little bit about the long-term, I don't know, the long-term path of this population, the hybrid population. Like,
Oh,
is
yeah.
it going to persist, or is it doomed, or what?
Yeah, I think they're going to, I mean, we already see other hybrid populations that have kind of collapsed into, that they seem to have more or less sorted the incompatibilities out. Like there's not strong selection happening because they've already fixed the compatible pairs of either thing if you don't have new migrants incoming. So I think that they'll keep being there. And actually, there's a different wing of the lab's work that's like, is this adaptive in some cases? Because there's thermal adaptation along these elevational clines. And it's like, oh, well, are the hybrids more fit at intermediate elevations than either the parents? Some preliminary evidence of yes. And so I think the hybridization is not the problem. I'm worried about them more just from a climate change and pollution perspective. But I don't think, I think the hybridization, there is a sort of like load that happens in the population because of that, but I think it sorts itself out quickly enough that I wouldn't expect that to be the thing that extincted the populations. Yeah.
I guess a follow-up is that, could you, in the lab, take one of those hybrids and pair them with any of the parental species that they recognize?
Oh, yeah. So that in terms, the hybrids are, and this is part of why we think these zones, there's a ton of mate choice work in Xiphophorus. It was like it was studied for cancer and for mate choice. On the mate choice side, someone showed a while ago that the hybrids are on average, extremely like permissive. They show less preference than either the parents. And so once you have some hybrids, that creates like a bridge for the ancestry to flow between the two if the parents are willing to mate with the hybrids. So yeah, we definitely, and honestly, it's the mate choice biology is really interesting because it's a little bit oversimplifying, but it's not that oversimplifying to say that the females are doing all the choosing. It's really about like the female's perception, because in the wild, you will see the males like attempting to court things in a different genus. They're like, it's a fish. It's like, so it's really about the female's perception. And I think there's cool stuff to be done with like this pheromone biology, because there's some preliminary stuff about the pheromone production. That seems like it might be a relatively like Mendelian trait. And so you could imagine hybrid, like a mismatch in the hybrid females between the pheromones that are being produced and their preferences for them. So that's a whole other cool wing that could happen someday by somebody else. Yeah. Any other questions?
Let's thank Ben again.